Okay, this is chapter 12, recombinant DNA technology. We'll talk a little bit about biotechnology, some different methods that are used. It sounds like this is rocket science, but it's not really. It's really about taking things that we understand from bacteria and sometimes a little bit from bacteriophages and applying it in test tubes and automating it. So the first thing we're going to go over is that the genetic codes we have and that we first learned about using E. coli are similar in all cells. All cells use DNA to store their inherited traits. They all use a series of RNA steps to convert those traits into proteins. They all have the same codons, and those codons code for the same amino acids that are used to make proteins. All cells use DNA polymerase to copy their DNA. And all cells can manipulate DNA with other enzymes as well. And so we've isolated those enzymes. And so these processes, if we purify the enzymes and put the enzymes and DNA in a test tube, they will work exactly like they work in cells. And we can even automate this stuff. And so, I've talked about how DNA is copied. I should also point out that uh, enzymes and probes recognize specific sequences of DNA. And so if you make a probe, you can have it bind to a specific sequence of DNA in a mixture of DNA sequences. And you can fish it out, and that binding will be stable. And so this means that we can find specific sequences that we want to cut or that we want to save. And these are really, really important. An example of this are restriction enzymes, that they recognize specific sequences of DNA and cut them. We found these in bacteria. They use them as a defense against viruses. And they often cut them with these staggered cuts, or what they call sticky ends. And we have other enzymes that can put these things back together. These are DNA ligase that heals cuts in DNA. It's used in copying DNA. It's a helper for DNA polymerase. And so these are really important things with joining pieces of DNA together. And so that's really important. Also, we use plasmids and phages from bacteria and viruses to move genes from one cell to another. That's what these things are designed to do. And so they're easily incorporated into a cell's chromosome. So knowing these types of things, in the 1970s, scientists recognized that you could combine DNA from two sources and probably stick it in another cell and get that cell to make a protein that it normally didn't make. And all you needed to do this was your source DNA that you could cut and put into either a plasmid or a phage, and then heal it with DNA ligase. Okay, and that's really recombinant DNA, or genetic engineering, is that you take DNA that you're interested in, and you cut it using restriction enzymes, and then you can heal it with DNA ligase. Okay, and if you put it into a plasmid, that plasmid is easily inserted into another cell. From that, you can then get that cell to produce the protein that it never made before, that came from your source area, your DNA of interest. And so you can get plants to make proteins from bacteria. You can get bacteria to make proteins from humans, like growth hormone or insulin. You can do all sorts of things with this, and this has turned out to be really, really useful. Recombinant DNA allows us to produce insulin so that we can use that to treat diabetes. And so we took a pancreas cell, we cut up the DNA with enzymes, And we identified fished out of there the insulin gene, and then we used the same enzyme on a plasmid and cut the plasmid with that enzyme and then healed that up with DNA ligase and then put it into bacteria. That bacteria is now grown in a tank, and pure human insulin can be isolated from there very easily. And those bacteria now have that gene forever, so they'll keep producing insulin as long as you feed them sugars and amino acids that they need. And so it's a very cheap way to produce very, very high quality insulin. You can also get the production of clotting factors for treating hemophilia, a bleeding disorder. You can produce growth hormone, and you can use that to treat dwarfism. You can produce an immune protein called interferon that helps people's cells recognize and destroy cancer cells. You can make vaccines this way. Many, many other ways things can be made this way. It's inexpensive and it produces very high quality materials. Another major application of this is you can insert genes for from a bacteria, Bacillus thuringiensis, that produces a toxin that's toxic to insects but not humans. And you can insert that into corn, which is Bt corn, and that corn now produces a protein that's toxic to insects but harmless to humans. Essentially, it produces its own insecticide. So you no longer have to spray the fields, you no longer have to kill insect predators. You can just wait till an insect feeds on that corn plant and then it will be killed by that toxin. In another case, we've inserted a gene for resistance to a herbicide, a broad spectrum herbicide called Roundup, into corn plants. And again, in that case, the corn produces its own resistance gene, so it resists that herbicide. But that herbicide kills all weeds, so very, very effective. Instead of having to spray multiple different types of herbicides, we only spray one. The corn is resistant to it. So that's genetic engineering. There's a couple other really simple and important applications of DNA technologies. One is called the polymerase chain reaction, or PCR. And here we use an enzyme from a hot springs bacteria. It's DNA polymerase from Thermus aquaticus, so they call it Taq polymerase. And we can copy DNA that we want to copy. So we can make unlimited copy samples from DNA. So all we have to put in is the source DNA, our enzyme, and some primers and nucleotides. And then this machine called a thermocycler will cycle at between temperatures that the enzyme works and which DNA separates. And so you'll get single-stranded DNA, and then you can allow the enzyme to work. And so this this cycling happens very quick, in a minute or two. So in a couple hours, you can produce billions of copies of source DNA for analysis. So you no longer need to grow organisms in the lab necessarily to identify them. You can identify samples to species or to individual from very, very small amounts, essentially one cell copy. Similar enzymes are used for DNA sequencing. We use DNA polymerase, and we take our source DNA and some primers, and we mix in some nucleotides and then some labeled chain terminators. These are similar to nucleotides, but they cause the enzyme to release the DNA, so they'll end a copy sequence, but they'll end it each with a fluorescent label. And these have different colors, so we can tell what was at the end of that segment. So each segment is of a different length and will end with one of these 4 different colored fluorescent dyes. So we separate these by size and get a computer to analyze it. And so this allows us to sequence DNA from any source, and the machine will read it. So this is automated. So all the technician has to do is load her resources there and her sample DNA, and in a couple hours she'll come back and read the DNA sequence. And so this is really helpful for diagnosis of infections, for finding specific mutations, identifying cancer cells, all sorts of things like that. Another really good example of this is DNA profiling. We can identify individuals from extremely small sample sizes because we have sequence technique and uh, polymerase chain reactions, and we can use extremely small samples and identify individuals by a variety of techniques that identify unique DNA sequences. This was first started to do this in the 90s. A group of lawyers got together when they recognized these techniques, and they started to raise money to re-examine evidence that was used in many cases. And this group is called the Innocence Project. And over about 30 years, they've re-examined hundreds of cases. They've overturned over 350 convictions. Okay, they've where the DNA evidence specifically excluded the person that was convicted. And then they've been used that same evidence to get over 140 new convictions where we can identify who left a DNA sample. So this is very important in cases of like murder where there's blood evidence from the victim or the perpetrator at the crime scene. Very, very important in sexual assault and rape cases where there's often a sample of the victim's and the perpetrator's DNA. And this has been a revolution in our justice system. In Illinois, over 20 years, we executed 12 people who were sentenced to death for capital crimes. Okay, in just the years between 1990 and 2000, re-examining those things, we overturned 13 convictions. That's why we don't have the death penalty in Illinois. We recognize that, okay, there's lots of these convictions are faulty, and so we re-examine that, and we don't do that anymore. So really, really powerful techniques, new forms of evidence that can be used in lots of different ways in agriculture, in medicine, and in criminal justice. So, since DNA is the same in all cells and has the same function in all cells, we can use enzymes and cell chemicals to find and manipulate DNA in test tubes, and we can automate these things. And this has allowed us to move genes, to identify genes, and to do lots of things with that. Okay, I'll stop there. I'll review this a little bit. There is a movie to watch on Moodle, so go to that and see if you can watch that.